All right, we're now going to turn our attention to NK cells. Um, NK cells are members of the innate immune response. And the reason for this is that they actually respond completely antigen non-specifically. There's no specificity to um, what they're looking for to kill. They are assassins, they kill on sight, and they're not very discriminating about it either. They're more like outlaws. Like they have like a code of their own that they live by, but as if you do not fall within the code that calls for mercy, you will be shown no mercy. And that's pretty much NK cells in a nutshell. However, I tend to talk about them at the same time that I talk about CD8 positive T cells. There are a lot of things about CD8 positive T cells and NK cells that are very similar, namely the way they kill. All of the killing mechanisms that I have laid out in the past for CD8 positive T cells are also used by NK cells. They secrete perforin and granzymes basically to punch holes in the membrane of target cells. They are capable of expressing Fas ligand. So they do the same things. They just don't have an antigen receptor. That's kind of the take home message. They're a CD8 positive T cell that is not looking for a specific antigen and doesn't express CD8. So instead of CD8, NK cells are typically identified by their expression of CD56. It's not on all NKs, but that's kind of the closest that we're going to get to it. And they're potent killers. The other thing that they're capable of doing is cytokine secretion. Specifically, they're able to produce cytokines like interferon gamma, which will activate macrophages, and the macrophage then will produce IL-12, and the IL-12 will actually further activate the NK cells. So they can work in kind of a symbiotic relationship with macrophages through this interferon gamma IL-12 exchange. All right, so I mentioned that like CD8 positive T cells, NK cells kill, but I didn't really say much more about that. NK cells, like I said, they have this kind of code, and the code is basically a balance. Do I have more signals that say kill, or do I have more signals that say don't kill? Like, basically, what is my motivation here? Are you telling me to kill or not to kill? Because I will do the thing that wins out. It's if you think back to ancient gladiator times, the ruler would either give a thumbs up or a thumbs down. Basically, the NK cell is giving the cell a thumbs up or a thumbs down. If the cell gets a thumbs up, it's going to live. If it gets a thumbs down, the NK cell is going to kill it. It's non-specific. So all of these are receptors and ligand combinations that exist on a variety of cells, and it just kind of needs to see enough of them. Most of the time, what happens is that NK cells have kind of activating receptors that provide kill signals, instructing the NK cell to kill the target cell. They also have these inhibitory receptors that provide stop signals, instructing the NK cell not to kill the target cell. The activation receptors are germline encoded, and they don't have any mechanisms of diversity. There's nothing that's going to say change about them. It's not like VDJ or anything that occurred with our TCR and BCR, okay? They're going to have a downstream signal transduction pathway that leads to death of the cell. Typically, an inhibitory signal is going to override an activating signal. So if I'm an NK cell and I'm making a list of kill and no kill, and I have one kill vote and one no kill vote, the no kills win. If I have 2 kill or 3 kill and only 1 no kill, likely the cell is going to be killed. It's kind of just that simple. The NK cell inhibitory receptors are necessary because many of the activation ligands for the activation receptors expressed by NK cells are constitutively expressed on a variety of cell types. So you kind of need to have a lot of no-kill receptors around because you're naturally going to have a lot of kill receptors around. So you have to use these inhibitory receptors carefully so that we don't unnecessarily kill our cells. The most important inhibitory receptor is self-MHC class 1. And if you think about it, this makes sense. Self-MHC class 1 is expressed on all nucleated cells throughout the body. So theoretically, every single cell in your body is supposed to express MHC class 1. This is the license that your cells need to drive around your body. Without it, when an NK cell comes up to interrogate it, the NK cell will assume it does not belong in the body and kill it. Alternatively, if it's not your self-MHC MHC molecule, the NK cell is also going to kill it. So the reason itself, and the reason I mention that is that, but this is really important in transplant. This is part of what our bodies would respond to during an organ transplant if our MHC molecules weren't carefully matched. Many pathogens like herpes virus and CMV and EBV and other tumors just in general, avoid recognition by CTLs by down modulating MHC class 1. So they decrease the amount of MHC class 1 expression. However, this makes the pathogen infected cell or tumor cell vulnerable to NK cell attack because they lack that MHC class 1 molecule. So if a cell lacks MHC class 1 or doesn't have self MHC, the NK cell is going to kill it because that's a really strong inhibitory signal. There are also these activating receptors. I've listed one here. The most important one is NKG2D. The ligands for NKG2D are kind of throughout the body, and they're just kind of a wide assortment of glycoproteins. I'm not really going to give you an example because you don't really need to know the specific ones, but those are the big deal ones. NKG2D is the big activating receptor. KIR is the big inhibitory receptor. KIR recognizes self-MHC. Okay, there's one other thing that NK cells really excel at, and that is antibody-dependent cell-mediated cytotoxicity. This is also known as ADCC. So you may have seen it referred to that way or heard me talk about it that way. This is only something that IgG can do. So this is a partnership between IgG immunoglobulin and NK cells. In ADCC, what happens is you get an infected cell that produces viral proteins on the surface. So for example, if we look down here, I want you to think of some of the herpes viruses again. Okay, so let's say we're dealing with HHV1. Okay, all right, so a person gets HHV1. They get their cold sore outbreak, right? Then it goes latent for a while, and then, years later, they get another outbreak with HHV1. All right. So HHV is able to infect those mucoepithelial cells. There are other herpes viruses that can actually infect phagocytes, but this kind of works no matter what. So either way, the virus gets into the cell. And then what happens is, and we're actually going to look up here, the virus gets into the cell, and then as the virus is being transcribed in the nucleus and then eventually will be packaged into virions, there are going to be viral particles on the surface. Now, some of these could be part of creating that virion so that it can be released. Some of those are just, they get expressed on the surface even during like latent phase or early replication, not so much latent, but early replication. So you have these surface antigens. Now, if the first time you made antibodies to it, antibodies are now going to start sticking to your own cell because this antibody is specific for this viral antigen. So the IgG antibody starts coating a self cell, okay, as if it's foreign. Your body doesn't really care. What it sees then is that you've got an antibody-coated object. So what's going to happen is the NK cell has a receptor for IgG. It's called Fc gamma receptor 2A. Now, let's just break this down briefly. So if this is my antibody molecule, right? This up here is my FAB fragment. This down here is my Fc fragment. So what this means is that this is a receptor for the Fc portion of IgG. That's all that means, okay? It's also known as CD16. But basically, the NK cell has this Fc gamma receptor. It's going to bind the Fc portion of the IgG molecule that is bound to the surface antigen that is produced by the virus. And the NK cell is going to go, oh, this cell must be virally infected. Kill it, die. And that's pretty much the whole thing. And it's very, very successful at that. So ADCC is a really potent way that NK cells can kill, and it actually is kind of a crossover to the adaptive immune response. Okay, so that's pretty much it. We've got macrophage activation. Remember, that's through the exchange of interferon gamma from the NK cell to the macrophage, and IL-12 from the macrophage to the NK cell. You can do direct killing of cells. That's going to be through perforin or granzymes. And remember, this direct killing of cells is going to be based on a combination of kill, no kill signals. The most important inhibitory signal being self-MHC, and that is recognized by KIR. And then the third mechanism is ADCC, which only works with IgG, and it works because the NK cell has an Fc gamma receptor.